Hello everyone, welcome to the channel Microbial World. Today let's learn about generalized transduction process. Transduction is a process by which genetic material is transferred from one bacterium to another by a virus. In simple terms, we could say that it is a process where bacteria gets new genetic material from viruses. It is a form of horizontal gene transfer because transduction transfers genetic material from one bacterium to another, often between unrelated bacterial cells. It is significant in the spread of antibiotic resistance and other genetic traits among bacteria. There are two types of transduction: generalized transduction and specialized transduction. In generalized transduction, any part of the bacterial DNA can be transferred, whereas in specialized transduction, only specific parts of the bacterial DNA near the virus's integration site are transferred. So let's discuss more about generalized transduction. As we have already said, generalized transduction is a process by which any bacterial gene can be transferred from one bacterium to another via a bacteriophage. And what are bacteriophages? These are viruses that infects bacteria. Phage infection. A bacteriophage infects a donor bacterium and injects its DNA into the bacterial cell. Degradation of host DNA. The phage DNA takes over the host machinery to replicate itself and produce new phage particles. During this process, the host bacterial DNA is often fragmented. Assembly of phage particles. As new phage particles are assembled, some of them mistakenly package fragments of host bacterial DNA instead of phage DNA. These phages containing bacterial DNA are known as transducing phages. Release of phage particles. The host bacterium eventually lyses, releasing both normal phages containing phage DNA and transducing phages containing bacterial DNA into the environment. Infection of a recipient bacterium. A transducing phage containing the donor bacterial DNA infects a new recipient bacterium and injects the bacterial DNA. Integration of donor DNA. The injected bacterial DNA can undergo homologous recombination with the recipient bacterium's chromosome, integrating into its genome and conferring new traits to the recipient. Thank you for watching.